Hello, everyone, and a very warm welcome to all of you for joining us today. My name is Anuja Bhardwaj, and I will be your moderator and host for today's session. Before we begin, I would like to share a few quick housekeeping notes. This webinar will run for approximately 50 minutes, followed by an interactive Q&A session at the end. We encourage you to make this session engaging by submitting your questions or comments at any time using the chat box located on the right-hand side of your screen. We will address as many questions as possible during the discussion segment. Please note that all participants are currently in listen-only mode. Today's webinar is being recorded. The recording will be shared with all registered participants after the session. To get us started, our session today is on evolving immunogenicity bioanalysis for vaccines and biotherapeutics. Immunogenicity assessment remains a critical component in the development of both vaccines and biotherapeutics. Yet, the underlying strategies and regulatory expectations differ significantly between these modalities. This webinar will explore the evolving landscape of immunogenicity bioanalysis with a focus on the differential bioanalytical approaches required for vaccines versus therapeutics and biologics. To help us navigate this, we are pleased to have our speaker for the day, Dr. Kedar Puran Patre, who is PhD in molecular biology from the Indian Institute of Science and brings over 28 years of experience across academia, pharma, and CROs, specializing in bioanalysis of small as well as large molecule therapeutics. He has authored 50 plus publications, patents, and is a recognized scientific leader, editor, speaker, and founder of the Bioanalysis Group of India. Currently, as Director of Bioanalytical Operations at Erogen Life Sciences, Dr. Kedar leads bioanalytical solutions supporting regulatory submissions. With that, I would like to invite our speaker, Dr. Kedar, to take this session forward. Over to you, Dr. Kedar.
Thank you so much for the introduction, Anuja. And good evening, all. I'm thankful that you're attending this webinar. As vaccine platforms and biotherapeutic modalities are changing, so are our challenges to address the immunogenicity for them. So in next 45 minutes, I'm going to share with you how the immunogenicity assessment has moved from antibodies to understanding the integrities of how immune cells interact with these therapeutics. To begin with, I'll just give you a brief introduction of our organization. Our science, we are a complete CRO CDMO where we support your programs and your science moves forward with our solutions. We have a simple purpose that in every molecule there is a possibility of better health. From our leadership to staff, our ethics determine how we do what we do for our customers, for each other, and for every stakeholder at Aragen. We deliver solutions from concept to clinic and concept to commercial. We deal with small molecules, peptides, protacs, oligos, ADCs, and biologics. And to deliver the solutions from the inception of your project all the way till the launch and phase for manufacturing, this is what we achieve it through. We generate more than 70,000 molecules every year. We support more than 250 plus active clinical programs, including 20 late stage commercial programs. We support more than 100 plus, we have supported more than 100 plus INDs, and we are partnering for 4 commercial new biological entities. We are supporting 400 plus customers with our 4,000 plus scientists with the highest number of PhDs in the CROs, CDMOs in India. We have 6 world-class campuses, 5 in India and 1 in California. We focus on speed, quality, safety, and talent base. At Aragen Biosafety Solutions, we are your partner for regulatory bioanalysis. We start partnering with you from the beginning when you have a molecular concept. We synthesize those compounds for you, including small molecules as well as large molecules. We support them through formulations, CMC bioanalysis. And once you have a lead candidate, we support you through GLP safety assessment, including toxicokinetics and immunogenicity assessment. Further, as your program moves into clinical studies, we support you through clinical bioanalysis from phase 1 to phase 3 for your samples. And even after your molecule is launched, we help you to understand the efficacy of a molecule through therapeutic drug monitoring. We help you for efficient transition from preclinical to clinical, saving cost, time, and resources. In today's presentation, we are going to cover these topics. We are trying to understand immune responses relevant to vaccines and biotherapeutics, emerging bioanalytical approaches for modern therapeutic modalities, and the regulatory expectations, key challenges we face, and the future directions in immunogenicity assessment. To begin with, let's start understanding what is immunogenicity. Immunogenicity is a substance ability to provoke immune response in the study. So foreign molecule is called an antigen. This antigen interacts through various immune pathways and activates T cells and B cells, which lead to formation of antibodies, which support protection from the antigen. Immunogenicity is essential for predicting, minimizing risks, and guiding safety and efficacy of both vaccines as well as biotherapeutics. If you look at the immune system closely, it is divided into three parts. The first part is innate immune system. So as soon as the antigen comes in contact with the receptors of our immune cells, they secrete cytokines and then work through complement mediated pathway. If the antigen is presented to the T cells, then they activate through various cytokines, they activate other T cells and B cells, which is called cellular immune response. And later on, it translates into humoral immune response, where the B cells synthesize antibodies which give protection. Now, whenever we talk about immunogenicity throughout this presentation, we are going to focus on clinical consequences. What happens if there is an immune response? If it has no impact or on the PK as well as efficacy and safety, it is not. We don't have to worry about it. But the immune response can vary from there all the way to mild hypersensitivity, pseudo-allergic reactions, anaphylaxis, loss of efficacy, severe tissue inflammation or damage to autoimmune phenotype. And these are the signals that we need to monitor throughout when we are administering vaccines or biotherapeutics. So the real challenge is identifying which response becomes clinically meaningful. This cartoon shows the intricacies of immune response. When the antigen comes into the milieu, it is presented by a dendritic cell called as antigen presenting cells, and then it moves through T cells and B cells to create antibodies. The initial part of the response is innate immunity, which is a rapid response, and it is transient. While the adaptive immunity is slow response, and it persists and it gives long-term protection. Now, to assist this, there is a lymphatic system which hosts the cell and allows these interactions, as well as it also helps these cells to move around our body. So the toxicity arises from overactivation or misdirection of these immune pathways, and early bioanalysis prevents immune-driven safety risks. Like every coin has two sides, immunogenicity has a dual nature. In vaccines, it is protective. And as we know, for the vaccines, we inject the attenuated pathogens or their proteins inside the body. It activates our immune system and it generates antibody. So whenever the pathogen or protein enters in our body for the second time, these antibodies are ready to interact with them and get rid of them. So this is what is our goal, that we need a protection from a pathogen or a foreign protein. While in biotherapeutic, it becomes challenging. When we give the a biotherapeutic to a patient, patient generates an antibodies, and if these antibodies have an ability to neutralize, they cause rapid clearance of your target molecule. Now, if your molecule cannot sustain in the body for sufficient time, you start losing efficacy, and also it leads to side reactions to your biotherapeutic. We will go through vaccines to begin with, and then we will see our challenges with biotherapeutics. So vaccines are evolved from different classes. In Aragen Biosafety, we have been working over 2 decades on various classes of vaccines. And the list that I have shared here are the actual vaccine studies which were conducted in our facility, including their immunogenicity assessment. Vaccines have started from inactivated pathogens, then live attenuated pathogens. They moved further to protein subunit vaccines, and then virus like particles. Now, during COVID time, we came up with new modalities like virus vector vaccines, mRNA, and DNA vaccines. Now, these vaccines, all of them act through a common pathway for generating the immune response. Once the vaccine enters into a system, it's normally through injection. There are dendritic cells which recognize them as a foreign particle. Now, these dendritic cells move into our lymph node and our lymphatic system, where they present the part of this vaccine and become antigen presenting cell. These antigens are recognized by our MHC receptors on the T cells, and then the T cells activate B cells to form antibodies. Initial response is IgM, and then the antibody matures to IgG, which gives you prolonged protection. Now, one thing we should notice that there is a red curve which is for inflammation, and there is a green curve for immunogenicity. So that the same players have a tendency to also cause inflammation. And since vaccines are given to an healthy individual, it becomes crucial to control the inflammation and direct this pathway towards immunogenicity or to immune response. As the vaccines have evolved, there are different ways of the way they give immune response. If you go to inactivated vaccines, they normally raise only antibodies. And so the assays that we do is binding assays for antibodies, which is a gold standard for all vaccines. And along with that, we have to demonstrate neutralizing activity. For attenuated vaccines, in addition to humoral response, you also get cellular response. So we conduct ELISpot or flow cytometric based assays for T cell responses. Recombinant vaccines usually give specific humoral response, and here functional assays for neutralizations are critical. Conjugated polysaccharide vaccines show T cell dependent responses and along with the robust IgG response. So for here, along with the subclass assays for IgG1, IgG2, one has to do functional assays like opsonophagocytic assays or serum bactericidal assays. Parasitic vaccines, I'm going to show you a case study in a while. They are complex. There, the antibodies cannot enter inside the host cell which harbors this parasite. Hence, the cell-mediated immunity becomes critical, and we have specialized assays for cell-mediated immunity. Viral vectors give you robust cellular response, a strong T cell activator, and humoral response. So again, here we do ELISpot for T cell activation. We look at intracellular cytokines as well as antibody neutralizing assays. Adenoviral vectors have a risk of pre-existing antibodies if you have already been exposed to vaccine. So your pre-existing antibody screen becomes critical. And mRNA vaccines give very strong cellular and innate immune response. Hence, that needs highest assessment of immunogenicity. We do neutralization assays, ELISpot for T cell response, and besides that, we also look at cytokine profiles because that innate immunity can cause inflammation. So the vaccine class dictates the immune readouts that are required for safety and efficacy evaluation. Now, these vaccines don't work alone by themselves, but they need a protein called as adjuvants. Adjuvants also have ability to induce innate cellular and immune response. Adjuvants amplify immune response by stimulating the innate sensors and enhance antigen presentation. Here is a list of adjuvants. Most of the responses to adjuvants are transient and they give us the side reactions like local pain, fever, and surge. However, it is always important to monitor the immune response to adjuvant. Hence, whenever we conduct any safety assessment study, we have a separate group of adjuvant where we monitor whether they go towards inflammation by looking at acute phase protein response. The new class of vaccines, and I do call them vaccines because this was presented in the vaccine conference last year as well as this year, are prophylactic antibody vaccines. These are the antibodies are directly given to prevent infection and immediately give passive immunity to high risk populations. The best example is RSV antibodies, which are given to infants who are at a high risk of having an infection. It was also tried in COVID-19 and has been explored for Ebola infections as well. So the challenge is now the vaccine itself is an antibody. So first important thing is one needs to measure how long does this antibody stay in the body, how well it is distributed, and when is it cleared. Besides being that, since it is an antibody, there's a risk of anti-drug antibody formation, and that can neutralize its activity. This has to be supported with functional assays to show that the antibody has an ability to control the infection. And the most critical factor is one has to demonstrate that the antibody stays in the body sufficiently for a long time and gives protection. Bioanalytically, the challenge that we face is that there are a lot of antibodies in the body. So one has to do very sensitive assays where you can differentiate them from endogenous background. The vaccine serology is changing and our challenges are increasing, and also regulatory expectations are increasing. As I said, it is given to the healthy individuals, hence the regulatory expectations are highest for vaccines compared to any other therapeutic. The critical challenge that we have is that getting the critical reagent, especially the positive standards, which are made from hyperimmune controls, when the trials long very last. Getting them consistently and with the consistent activity is really challenging. For looking at the background, now the regulators have moved and they want you to really look at the individuals which are not, whom they are not given vaccines. And like biotherapeutics, they want you to find out the background to confirm that your response is really true and about the background. A WHO expects that your assay is harmonized so that it can be reproducible in any geographical condition. From in modern vaccine bioanalysis, what we see is that there is a shift from broad serology to validated regulatory assays, and now our matrix has changed from serum to very complex matrices like mucosal secretions. For RSV, the saliva and mucosal secretions are used to look at the antibody titers. The platforms are moving to multiplexing as it's expected that your assay should not only measure antibody subtypes but also multiple antigens, multiple serotypes simultaneously. And COVID is an example where we have seen that the antigen was constantly shifting, and that's why it was critical that your vaccine supports all serotypes that are prevalent at that time. For bacterial vaccines, it's very critical that you have to demonstrate functional assays. Whenever we have worked with bacterial vaccines like meningococci vaccines or pneumococcal vaccines, we have supported it with opsonophagocytic activity for pneumococcal vaccines and serum bactericidal activity for meningococcal vaccines. Regulators are very critical. Unless you show this efficacy in functional assays, the vaccines are not approved for the public. This is the bioanalytical toolbox we use to do the analysis for immunogenicity. ELISAs are done to identify the binding assays. We do neutralizing assays using cell-based or in vitro systems to show the functions of of these antibodies. ELISpot, as I've shown, is used to identify cytokine secreting cells and cell-mediated immunogenicity. MHC tetramers assay is used to look at the antigen specificity. Multiplex cytokine assays, as I shown earlier, is used to look at different serotypes and antibody subtypes. And immunophenotyping becomes critical if your vaccine is having an immunomodulatory activity. The platforms that we use are we have these citation systems which can do multiplet reader as well as it is also an ELISpot reader. MSD is the most sensitive system and that is used for multiplexing assays. Luminex platform also helps us in multiplexing for doing vaccine immunogenicity, and FACS is used for the cell-based analysis. I'm going to share a case study of vaccines just to show you the intricacies of evaluating immunogenicity against vaccines. Now, this is a case study where we had a live attenuated parasite vaccines. And as I mentioned earlier, parasites like Trypanosoma, Leishmania, Plasmodium are intracellular, that is, they survive inside the host cells. And the antibodies cannot reach them, hence, humoral immune response is not sufficient. In this case, it's very important to have a cell-mediated immunogenicity, which is driven by a Th1 type of response, driven by interferon gamma and cytotoxic T cells. So the way we had demonstrated it in a preclinical model, initially we looked at the antibody titers, and we looked at the antibody subtypes IgG1 and IgG2A. Now, IgG2A drives the Th1 based immunogenicity, while IgG1 gives a normal humoral immunogenicity. And we looked at the ratio of IgG2A to IgG1, and in all the instances where the vaccine was given, we saw that the ratio is greater than 3. That confirms that the vaccine is going through a Th1 pathway. Another assay we used was where we took the splenocytes from vaccine-treated animals. We incubated them with vaccine antigen and then looked at interferon gamma and IL-10. And we saw that the concentration or the induction of interferon gamma was higher than IL-10, again suggesting that vaccine goes through the expected Th1 driven pathway. So this just shows that the immunogenicity assessment for safety and efficacy of vaccine is more than ADA. You need to support it with specialized assays for the progression of the vaccine molecule. Now we will move to the immune response to biotherapeutics. The source of immunogenicity risk of biotherapeutics is not single. It can be product related, the sequence, aggregates, impurities, post-translational modifications, formulations, or route of administration. It can be patient related, the disease state of patient, their genetic makeup, their immune status, their prior exposure, and concomitant therapy. And these are all factors all have to be considered when we do immunogenicity assessment for biotherapeutics. Treatment-related factors like dose levels, durations, route, intermittent dosing, or treatment interruptions. Now it becomes challenging since there are multiple factors to address while assessing the immunogenicity of biotherapeutics. We will walk through it class by class. This is the general overview for a protein therapeutics which starts from peptides, peptibodies, hormones, pegylated hormones, fragment antibodies, monoclonal antibodies, receptor chimeric antibodies, to all the way antibody drug conjugates. The most critical immunogenicity risk is generation of anti-drug antibodies, which can alter the therapeutic exposure and neutralize the therapeutics. The classical assay that is recommended by both FDA and EMA is ADA screening tiered strategy. Now, this is used throughout for all biotherapeutics, and I'm going to walk you through with additional assays that we go with each class of biotherapeutic. To begin with, I'll just describe what is an ADA screening tiered strategy. In this strategy, you first establish a screening cut point using 50 LD patients, multiple runs, that is almost 6 runs. So you have 300 assay points, you do statistical analysis, and you find out that this is the background of my assay. Now, any sample that is above that background is called a screening positive. Once you have a screening positive sample, you take it for a confirmatory assay. In confirmatory assay, you perform assay in presence of excess of biotherapeutic. And if you can demonstrate specificity of your molecule, then you call that this sample is positive. Now, positive samples are then further analyzed for their titer, which shows the magnitude, neutralizing activity, which shows their effectiveness, isotype, and epitope mapping, depending on the class of therapeutics. I'll just walk you through the different classes and modalities which are currently into the clinics. Peptides have recently become a hot favorite because now we have oral peptides. and they have shown very good efficacy and relatively less immunogenicity risk. However, it depends on the homology of the peptide. Here is an example for GLP-1 receptor agonists. If you have a molecule which has a very different sequence than the endogenous molecule, like exenatide or trizapatide, we can see that due to sequence divergenicity, the ADL incidence is very high. It goes from 30 to almost 70%. While for peptides like semaglutide or dulaglutide, which are 97% similar to GLP-1 peptide in the body, the immunogenicity risk is less than 2%. So the triggers for peptides are not only the sequences of the peptide, but also the impurities which are generated during their synthesis. There are special impurities which induce innate immune response, and these are called as innate immune response modulating impurities. Besides that, when you do semi-synthetic peptides, the host cell proteins are also a concern. So the assays we do, as I said, a gold standard ADA screening tiered strategy is done. Along with that, one performs in vitro MHC or T cell based assay to predict immunogenicity, epitopes, and T cell activation risk. We have specialized in innate immune response modulating impurity panels. Against that, one has to see if there are any IRM related peptide impurities. And one also looks at host cell protein analysis to confirm that you do not have any additional triggers in the molecule that you're administrating. Another very interesting class of molecule, and it is really a boom in the biological sciences, is enzyme replacement therapies. Now, these enzymes are not produced by our body, they are given exogenously, and we do not have any endogenous counterpart for it. Since these are foreign proteins, the body does generate antibodies against them. And if these antibodies turn to be neutralizing, then the enzyme is cleared very quickly from the system, and hence you do not get the desired benefit. And best example is asparginase. So for a patient, you start with equal asparginase. Over a period of time, the patient, this is used for the acute leukemia. Over the time, the body develops antibodies against equal asparginase. Then you shift to Erwinia asparginase, which is structurally very diverse but has similar activity. So as I said, the challenges or the immunogenicity risks are binding ADA formation, neutralizing antibodies, infusion-associated reactions, and anaphylaxis and hypersensitivity. So the assays that we perform in the lab are we do ADA screening tiered strategy, we look at IgG subclass characterization, and we also support it with T cell proliferation and cytokine profiling. Another class of molecules which are coming up in a big way are these multispecific antibodies, like bispecifics and trispecifics. Now, these are specialized antibodies. So they introduce, since they have multiple domains that bind to the target cells, they generate neoepitopes. Besides that, they also directly interact with the immune cells. So the challenges associated with them are higher. One has to not only look at the ADA detection, but one has to show whether there is domain-specific antibody, and that is mostly done by using confirmatory assays. One has to demonstrate that is there any neutralizing antibody activity, and when we correlate it with the pharmacokinetics to show if the antibody that is generated had any meaningful impact on the presence of or efficacy of the molecule. Besides that, since they directly interact with the immune cells, it becomes very critical to monitor the cytokines because you do not want the response to go towards inflammatory pathway, but you want them to stick to immunogenicity. And immunophenotyping also becomes critical as these are mostly immune cell engagers. Moving ahead, now we are into the world of antibody drug conjugates. It's a beautiful system where you can take a very toxic drug at low concentration and your antibody directs it to the cancerous tissue where the drug is released and then it penetrates inside the cell and kills it. So this is what is expected. However, while generating these antibodies, you modify the backbone of your antibody, and then that, along with the linker and the drug, creates again new epitopes. These become hotspots for immune reactions and immunogenicity. The consequence is that if you have antibodies which bind to them, then they do not allow the drug or the molecule to reach to the target cell, and hence you do not see the expected efficacy. Besides that, they form immune complexes which will give you infusion reactions and off-target toxicities. The assays that we conduct in our labs are ADS screening tiered strategy along with domain characterization. We identify epitopes uh, like by using various assays. We look at the neutralizing antibody assays, and at times even SPR is used to find out the binding kinetics. This is the latest molecule in biotherapeutics, which is cell and gene therapy. Now, this poses various challenges. In gene therapy or viral vectors, if you are already exposed to these vectors, your body will be having an antibodies. So, whenever you have any other molecule coming in the same vector background, the antibodies will neutralize it and will not allow it to express or show its efficacy. So pre-existing antibody screen is essential in this case. Now, when you go for gene therapy, you use proteins like CRISPR, Cas9, which are expressed in E. coli. Now, these are foreign proteins to our body, and our body generates antibodies against them. Besides that, even the transgene that is expressed is also foreign protein, and there is a likelihood of generating anti-drug antibodies against that protein. For CAR T cells, again, there is a risk of generating antibodies against the CAR receptors, and binding of those antibodies can actually clear the cells from the system. So as we see in CGTs, there is a risk of viral vector immunity, transgene immunity, cell therapy immunity, and innate activation. Now it's known that the DNA and RNA that we give to the patients has a binding ability to TLR, and it activates immune response. And if not controlled, it will lead to cytokine storm by releasing very excessive amounts of cytokines. So for this class, there is the highest assessment of immunogenicity that is done. We do classical ADS screening with tiered strategies for antibody detection. We look at cellular immunity by ELISpot, where we look at T cell responses to the capsid or transgene. We use flow cytometry to see the activated markers like CD69 or interferon gamma. We basically look at innate immune response as well, and cytokine profiling is suggested heavily along with the complement activations for CH50, C3, and C5a. Functional neutralization assays are also used to confirm that the antibodies, whether they have any impact on the clinical efficacy. To assess these immunogenicity risks, we use different platforms. I'll first walk you through the preclinical toolbox. So when a biotherapeutic is designed, an in silico epitope predictions are used to find out if there are any T cell binding sequence epitopes, so that can be minimized. Then we support them with in vitro and ex vivo human immune cell assays, where the PBMCs are taken, they are enriched for CD40, CD4, then they are challenged with the antigen, and then upon the second challenge with the antigen, they start secreting cytokines, which are monitored through ELISpot, where you can tell how many cells actually are responding to the antigen. And this gives you an early risk of incidence of immunogenicity. Besides that, we do product quality characterization, especially the aggregates aggregates and impurities. We do animal ADA observations, which has to be done with caution and context. Now, the golden question is: are animals still needed? What will the preclinical work can do for us? Will it really work well? And based on my experience, the answer is that yes, preclinical models are required to reduce uncertainty. What can they do? They can rank the candidate molecules by relative risk. They can flag high risk constructs. They can guide deimmunization. They can identify T cell dependent risk mechanisms and support clinical strategies. I'm going to walk you through one case study where I want to share with you how clinical models can be used to de-risk your molecule. So we were working with a chimeric receptor antibody. Now this was a fusion protein. It had receptors attached to the antibody backbone. Now, it's known that when you have fusion proteins or bispecific or trispecifics, they have a tendency to aggregate. So we got a particular formulation, and when we administered it intravitreally to the rabbit, we saw that the eyes were swollen immediately, and later on through histopathology, we could demonstrate that there was uncontrolled inflammatory response in this animal. And as inflammation and immunogenicity go hand in hand, we saw the ADA titers were tremendously high. Now, with this, we went back, we discussed, we homework, and we saw that whenever you have an aggregation of antibodies, it leads to the DAM signaling pathway. These DAM signals activate your antigen presenting cells. These hyperactive antigen presenting cells then activate B cells and T cells. And this strong B and T cell response leads to generation of large amount of anti-drug antibodies. So we worked with that team. They identified in vitro assays to identify the formulations where the aggregation was highly reduced. And those formulations were again checked in the rabbit model. We have confirmed that the AD titers were indeed very low, and also we did not see the inflammatory responses. Now, after that, that particular formulation moved to clinical study. So this shows that preclinical immunogenicity models can reduce uncertainty. They won't guarantee what happens into the human system, but at least such risks can be dialed out using the animal models. When we go to clinical scenario, the critical question is, okay, you have antibodies, but are they really significant, clinically significant? And the way that is addressed is we look at three different factors. The first factor is magnitude. How big is your response? And that is assessed by looking at the titers as well as the signal to noise ratio. Higher the titer is alarming, that it shows that your response is really high. Another aspect one looks at the time course. Is your how soon the response appears? Is it persistent or it's transient? And the third approach that one takes is looking at the maturity of this. What is the strength of your response? Does isotype switching happen? Do you see IgM followed by IgG? And most critical thing is, do you see increase in binding affinity of your antibodies over a period of time, which says that your response is persistent and also increasing. What is recommended, or we would recommend for the clinical studies, is that in this case, it's very critical to have a robust AD assay which has highest sensitivity and drug tolerance. One has to take sufficient samples throughout the treatment to understand the immune response. One has to collect samples prior timing to immunogenicity profile and accuracy. As I said, there are two critical factors and challenges for a bioanalytical group. One is the sensitivity of the assay, and another one is drug tolerance. Now, sensitivity for the assay is defined by regulators as 100 nanogram per ml, and this is preferably in pooled patient sera. Now, for small peptides like semaglutides, what we have seen that they generate very weak immune response, and they are very low affinity antibodies compared to large proteins. So it becomes standard in our ligand binding assay. We struggle with the sensitivity at the low nanogram levels because of two factors. Your positive control antibody does not have very high affinity, and another one is there is a background from the matrix that you are using. Another challenge with the peptides is that they may not present the stable epitopes. They reduce antibody that reduces antibody binding efficacy. And when we do a standard curves, since the peptides are in very high molar concentrations, the standard curve validate the plateaus at higher thresholds. So you do not get a very good linear responses like MAPS and other larger biotherapeutics. So the approaches that our team takes is we do pre-enrichment, we do acid dissociation, we do signal amplification with MSD platform. In our experience, we have seen that MSD gives the highest sensitivity and also reduced background that is required for looking at the immunogenicity of peptides. And I'm going to share a case study in a short while. Another critical factor is drug tolerance. That your antibody should be detectable in presence of maximum concentration of drug in the patients, and that definitely needs that you have to first separate your antibody or ADA from the drug, and that is done using approaches like PANDA and BEAD routinely in our labs. What do regulatory recommend for these? So the agencies recommend now that you should also look at IgM detection because that gives you early signals for immunogenicity. And there are critical challenges. We do not have positive controls for IgM, but our group has standardized certain protocols where we can generate and purify IgM positive controls, which can be used for setting up the assay. Besides that, IgG detection is a gold standard. One has to demonstrate that there is a class switching, and that it also becomes critical for certain biotherapeutics to identify the subclass of IgG. It's known that IgG1 gives better protection for vaccines as well as it also has a neutralizing potential in against biotherapeutic, while IgG4 and other IgG subtypes do not show such neutralizing activity. Hence, IgG subclass also gives you a hint or an indication that whether your antibody will have neutralizing activity or not. One has to do more clinically relevant, has to see the clinical relevance of neutralizing activity by looking at the pharmacokinetic parameters along with the immunogenicity assessment. Regulators also expect isotyping of antibodies where you distinguish whether the antibody just is binding or has a neutralizing activity. It supports risk assessment for safety and efficacy and provides mechanistic insights into the immune persistence. Immunophenotyping is also recommended for certain class of biotherapeutics, where the techniques like flow cytometry, ELISpot are used to profile T and B cell subsets. It identifies cellular drivers of ADA formation and also explains if there is any patient variability. This is highly recommended for the newer therapeutics like CAR-T, gene therapy, and DNA and RNA-based vaccines. If you look at the overall regulatory requirements when you submit your brochure, is EMA insists that you go through tiered approach, you go through screening, you go through confirmatory, characterization, and neutralizing assays. They recommend both IgM and IgG detection, isotyping, and immunophenotyping is done on case-by-case basis. The emphasis of EMA is on mechanistic understanding and answering the patient variability. FDA focuses more on clinical relevance of ADA. They want to know whether the response that you have got is just binding or it is neutralizing. They encourage functional assays to assess the impact of on the efficacy and safety. They have flexible approach but stress risk-based evaluation. Regulatory expectation is integrated interpretation and not just isolated reporting. I'm going to present a case study of a peptide, semaglutide. We have developed an in vitro assay for determining the three-tiered screening strategy, confirmatory strategy, as well as a cell-based neutralization assay. Now, while doing working with this peptide, as I said, the challenge was to get the signals about the background because the patient sera was diabetic patients and certain patients have taken GLP-1 like analogs earlier. So we have worked a special technique where we worked around minimum required dilution of the sera and then found out the right conditions where we could detect signals above background. Another challenge was sensitivity because, as I said, at 100 nanogram, the molarity of peptide is very high. And besides that, the antibodies that bind to these peptides are weak affinity. So we worked on it with various conditions, various kinds of positive controls, and we finally optimized an assay to a sensitivity of 90 nanogram per ml, which is close to the regulatory expectation of 100 nanograms. Confirmatory assay was very critical. We had to show that the antibodies have higher affinity for semaglutide versus endogenous GLP-1 peptide. And we show that the assays that we have developed can selectively show that the antibodies cannot bind to even 100x physiological concentration of GLP-1, and they bind to semaglutide at very low affinity. Neutralizing antibody assay was challenging. As I said, only 2% of patients turned out to be positive. So there were no positive controls commercially available. They had to be generated, profiled, characterized, and used for the assay. Again, getting a sensitivity in cell-based assay is always challenging because you have to work around with very fine conditions, and we have achieved the desired sensitivity. And we have also an assay which is highly resistant to the drug interference. So to sum up, I would close the presentation with these comments that for vaccines, immunogenicity is our goal. For biotherapeutics, it is a negotiation with the immune system. Bioanalysis has evolved from measuring if antibody exists to mapping the immune ecosystem around our product. From preclinical risk screening to post-marketing surveillance, immune bioanalysis is now a continuous feedback loop between the clinic and the lab. Bioanalysis is involved right from the beginning, like as I've shown, from the preclinical to all the way to clinical, and then even post-marketing surveillance. It is not a one-time event that you just conduct your three-tiered ADA assay and then you submit it to regulatory agencies. Now, agencies expect that you give the integrated picture of how your molecule behaves through interacts with your immune system, and that's why bioanalysis becomes critical to support the path of your molecule right from inception till clinic and even post marketing surveillance. So the goal has shifted from counting antibodies to understanding the impact. And we are designing, the question that we ask is that, are we just designing our assays to identify the antibodies or really understand their impact? Thank you so much for your attention. And now the talk is open for questions.
Thank you so much, Dr. Kedar, for such an insightful and informative session. We truly appreciate you sharing your expertise and these valuable perspective with us today. So we'll now move into our Q&A session, and I can see a lot of questions coming in. While we may not be able to cover all of them live, we'll do our best to address as many as possible. But please do send your questions, and if it is not addressed right now on live, we will do that, and our team will follow up with you via our email. So, just to start with the Q&A session, there is an interesting question, Dr. Kedar, that has come from Monal. So, it's how to correlate and interpret WBC count with immunophenotyping.
So in immunophenotyping, we do it mostly with the T cells. We do not look at the RBC or WBC counts. So the kind of assays we do, we get sera samples, which are does not have the cells that are required for analysis.
Okay, and someone has asked a question like, okay, so Arigen's expertise lies in which phase of drug development for regulatory bioanalysis?
So I think, Anuja, you're aware we are in every part of the drug development. We have our own cell proprietary cell lines where we generate those antibodies. Then we do the initial work in our laboratories. We do bioanalysis. We support you through your GLP talks as well as we support you through your clinical phases where the samples are shipped from clinics directly to us. We do analysis of pharmacokinetics as well as immunogenicity, and we support your filings with the CDC, SAO, as well as global regulatory agencies. And again, once your antibodies are there in your phase 4 as well, we support you identifying if the ADA prevalence is higher in the market, and then the dose adjustments can be done for the patients. So we are your partner at every step of regulatory submissions.
There is one more question like, so they have been asking on like, could you please elaborate on in silico or computational tools which can be used for prediction of immunogenicity these days? Like, what is the
Yeah.
current scenario?
Absolutely. So there are different tools coming up which predict whether your biotherapeutic can bind to T cells or MHC certain classes. It's just that as I've shown you that it's not a single factor. It also depends on the immune status of the host, the disease state of the host, as well as what else is there in your product, your excipients, the impurities. So there is no one single tool that is available currently to predict immunogenicity. These are just the preclinical tools at the moment. That in case if you see a higher titer, you can take your molecular sequence and identify if there are any classical epitopes, and then they modify and bring your molecule back. So at that stage, AI is quite in the initial stage for predicting immunogenicity. And since we saw that there are so many factors, so many players, it is going to be challenging to get a single or a very composite tool that can actually predict immunogenicity confidently.
Okay, yeah. That is there because now NAMs are also something which are coming into the picture, and how successful they are and not, that is still a question. So,
Absolutely.
Dr. Kedar, you show what exactly and how we like Erigen is supporting clinical bioanalysis. But then a question has come like in clinical bioanalysis, transport of samples is important, and in geopolitical situation, how you are managing the same.
Very good question. This is what every sponsor asks us. So generally, what is done, even we work with NHPs across, where the samples come from international laboratories to us. We give standalone bioanalytical services to all kinds of the modalities. So the way it is handled is you normally make an aliquots. You should at least have three aliquots, and they're shipped at different times. And you have to be very unlucky that even one aliquot doesn't reach you. Normally, at least two out of three aliquots will reach you, and the analysis can be done. So we are actually working with a lot of courier agencies who have an expertise of delivering products in time. So as I said, yeah, the option is to make aliquots and ensure that the deliveries are done at different times.
Okay.
Thank you.
Another question from Dipesh. I'll take this one. So, what is MRD and why does it matter? And how do you select the optimal dilution for that?
Wonderful. Good question. I want you to visit. I've started a forum called as Bioanalysis Group of India (BGI) on LinkedIn. I do explain these small concepts, right from which standard curve to use, what kind of kinetics, and again the basics of immunogenicity. So this is a very good question. MRD is minimum required dilution. So when you have your sera samples, it is full of antibodies. So if you just put it neat, you're going to get plethora of signals. You cannot control them. So you have to work on the dilutions, and the dilutions have to be such that your antibody should be detected, but the background has to be nullified. So that is what we do in our laboratories. We try various dilutions, various conditions, and we ensure that assay is sensitive to detect a specific ADA or antibody that we want, but the background is reduced. And this happens because when you go for small molecules, it's a very simple thing because your molecule you can precipitate a protein. It's not a molecule; it's a compound, and then you can use it for analysis. But your biomolecules are protein. You cannot separate protein from a protein. That is rarely done. You have to work along with the other proteins, and here dilutions come into picture. So minimum required dilution. MRD is minimum required dilution.
Okay.
Thank you.
Another question, like I'm also interested in this one. Like, how do pre-existing antibodies or prior exposure to similar products affect immunogenicity assessment and interpretation?
Yeah, this is also an interesting question. If you have pre-existing antibodies, you already have a lot of signals in your samples. So you need to have again a very sensitive assays where you can differentiate if anything has come up. So the assay strategy has to be critical, and also the time has to be monitored whether your signals are increasing from the pre-dose to the post-treatment. So that is how we do it. We look at the magnitude if it is increasing, we look at the strength if that is increasing, and we work around assay conditions. But yes, they
Okay.
are the assays are challenging, and having pre-dose antibodies increases the risk that your therapeutic may not work. So it will have a little
One,
bit.
yeah,
Yes.
okay. So one more interesting question, like, and it's a very critical one. How Erigen Biosafety support that pre-BLA meeting?
Can you just repeat the question? How harassment biosafety?
Support at pre-BLA meetings. BLA.
Okay,
Yeah.
the applications.
Biological. Yeah.
Absolutely.
Yes.
We are your partner throughout. As I said, even when you are doing a preclinical safety assessment, we are your partner in RCGM. We come and we work with you. We present the data that we have generated and we defend it. Same thing with Bele. Our team will be helping you with preparing your brochures. And also, if there are regulatory questions and you need us, we will support you in addressing them. And there is an instance we were working with one of the client for their novel therapeutic, and then there were queries from Health Canada, and we could address them. We could add the additional data that the regulators are required, and that molecule was then finally approved in Canada. So we have very good experience of supporting you through various regulatory agencies.
Given that like a lot of regulatory associated questions have come in, what I like there is one more question like what changes do you foresee in immunogenicity regulations, assay technologies, and data interpretation going ahead?
Okay, very good question. It's really a futuristic question. So what I see now, if you look at European Bioanalysis Forum or the discussions that are happening in our field, everybody is trying to make the assays more simple, more robust. So from doing now currently all the assays are done in duplicates. So singlet assays are going to take over. So that will actually reduce the efforts and also the requirement of samples. Another thing I had also discussed in earlier my presentation about hidden immunogenicity. So there are certain biomarkers which actually predict immunogenicity much earlier. So those will start coming in. So it's not just going to be an antibody, it's going to be an integrated approach where you look at your antibody, you look at your cell types, your cellular responses, you look at biomarkers, and then you make your assays simple and more robust. Also, at the moment, we have three-tiered approach. So, what is going to happen based on the discussions in WRIBs and other bioanalytical forums is that they're trying to just use signal-to-noise ratio for both confirmatory as well as the screening assays, and even predict titer based on that. So, the more data will be generated, and then your AI and your two tools will be used to analyze this data, and hopefully the immunogenicity pathway is just going to be looking at signal to noise ratios, looking at the biomarkers and T cell responses. A totally integrated approach is what will happen.
Okay, great, great. So there is one more question, which is like I think most of our clients need, like they are also interested in this question's answer. Anna Thomas is asking like why US client like us should consider Origin for biosafety.
Okay. We are doing a lot of IND studies. We are working with US clients. We have all the technical knowledge. We have people who have submitted their molecules when they work in pharma for the US agencies. And we have all the, you can say, infrastructure, knowledge, as well as experience to support you through your INDs. So definitely US and Europe clients they are already working with Aragen for production of their molecules. They can also work for biosafety and clinical studies along with us.
Okay, so there is one more question from Gajbara, and he is asking like neutralizing antibodies blocks semaglutide, and the result is that there is no adenylate cyclase activation and no cyclic AMP rise as well. So, what is the actual readout and why cyclic AMP?
Okay, now this is there were no commercial kits available, so there was a kit for bioassay where when anything binds to your GLP receptor, it will activate adenyl cyclase. These are all engineered cells; these are not natural ones, CHO cells, and the readout is luminescence. So you give a substrate which will convert your cyclic MP and then give you that substrate as an antibody which binds and glows. So the end point was luminescence, right? So that is the answer. The end readout was luminescence and using engineered cells.
So personally, I have a question for you, Dr. Kedar. Like
Mhm.
you have walked us through immunogenicity, bringing all the modalities which are currently hot and actually being studied, and most of the people they have in their pipelines those therapies. So my question is like, why immunogenicity will like? Is there any scope that immunogenicity ever be completely eliminated from biologic therapies, or like how it is going to be? Because every time immunogenicity is a problem and bottleneck in all of those therapies, even though they are very effective, but immunogenicity becomes a bottleneck. So how you will take this?
So the answer is it's very less likely that will happen because anything that is foreign, even the attacks that happen to us from pathogens or other challenges, will always be recognized by our immune system. It's our protection mechanism. And we do not see that happening. That is not our goal. Our goal is to reduce it. And it's not to eliminate, but predict, mitigate the response then manage it clinically so that it does not create other side effects or it does not affect your efficacy. So it's not going to go away, but it can be reduced, and the whole community is working around it to minimize it.
Okay, okay, yeah.
Thank you so
I
much.
was also expecting this answer, but was just curious if we have some kind of magic wand and it can just go and vanish.
Exactly,
Thank you
Stone.
all.
But I said there is no magic. It's a biology, and it has to happen its own way. It's just that we will come up with more smarter ways of dealing with it. And
Yeah,
actually, what we are seeing, as we go to complex molecules, like we have gone to cell and gene therapy, the risks have increased. If you start making something very similar to what is there in a body, then the chances, like I said, give an example of semaglutide, the chances are that the risk will be reduced. But whenever we become, we do experiments and we become very bold and come up with new theories, our body is going to react to it.
yeah.
Thank you so
So,
much for all the questions and your support.
yeah.
Yeah.
That
Is
is
there any?
all the time we have for questions today, and we have already extended the time to 2 minutes. If your question wasn't answered, or if you have additional queries later on, please feel free to reach out to us at info@origin.com. Thank you all for your active participation and for joining us today. Have a wonderful time ahead, and we look forward to connecting with you again soon for our next webinar on another engaging topic. So stay tuned.
Thank you.
Thank you, Dr. Kedar.
Thank you. Bye.
Thank you, everyone.